Okay. Um, Thank you for staying, so long, and for this pitch-up session. And first, I would like to thank the organizers for giving me the opportunity to communicate with you our work, which is preliminary, but we think very promising towards translation and towards meeting a high societal challenge, that of primary brain tumor, and namely glioblastoma. So what we have tried to do here is to evaluate the spatiotemporal distribution of a certain type of nanoparticles, theranostic nanoparticles, silica-coated, dye-doped, machemite nanoparticles in both normal brain and orthotopic glioblastoma models. And our major goal has been to see to what extent we can really breach the castle walls, because getting through the blood-brain barrier and making all the way to the brain, and not only to the brain, but to those particular areas of the tumor which the bad genes are located, it's a major challenge. And we hope to see more and more projects funded by European Commission in order to meet this, societal challenge. Well, a few words before going to our vision and actual mission. We work on primary tumors, malignant gliomas. I'm using here the old type classification because as of June 2016, WHO has classified primary brain tumors based on genetic and molecular data. But for the purpose of this particular presentation, I would rather stay with the traditional classification where we separate brain tumors, gliomas grade 1 to 4, grade 1 low malignancy, grade 4 very highly malignant tumors. And these tumors have very dim prospects for long-term survival. As we speak today, with all efforts and all money spent, median survival is about 14 months. And I've always focused on histologic and functional classification. If you see here, we speak of pilocytic astrocytoma and diffuse astrocytoma based on the functionality and the integrity of the blood-brain barrier. And as the tumor progresses, we have disruption of the blood-brain barrier in anaplastic astrocytoma and glioblastoma multiforme grade 4. So, in having said this, our vision has been to optimize the effectiveness of targeted therapies in gliomas, which are the most malignant brain tumors and the most malignant human tumors ever. Because here you have a deathly sick cancer patient and a very debilitated neurologic patient at the same time. So, need to minimize recurrence and improve overall survival for patients with a currently fatal and economically unviable disease. So it's a major societal challenge. And our mission has been to develop theranostic drug delivery systems capable of maximizing the bioavailability of target-specific therapies at the tumor-propagating areas, not any tumor areas, but the tumor-propagating areas, and assess their ability to challenge the blood-brain tumor barrier, specifically targeting distinct tumor cell populations, those are the cancer stem cells, and ultimately reduce the tumor burden. So the medical issues to address is the unexplained drug resistance we face in the management of glioblastoma multiforme. I'm glad we have already heard of precision medicine and precision nanomedicine, because here are several characteristics underlying and dictating a precision approach, which is the spatial phenotypic heterogeneity within the tumor, also accompanied by genotypic differences. So, within the same tumor, we have hypoxic and necrotic versus more normoxic regions. We have more proliferative versus more quiescent regions. We have very vascularized versus more infiltrative regions. And of course, we have in regions non-biopsied. So we face mosaic amplifications in a subset of gliomas and molecular subtypes of glioma stem cells. So the real challenge here is the lack of successful delivery of therapeutics across the blood-brain barrier and the tumor microenvironment and the extracellular matrix. So the parameters under consideration here as the blood-brain barrier breaching potential of any candidate target therapy. And if you follow me here, in the core of the tumor, we have disrupted blood-brain barrier, So any kind of drug can pull there, but this is useless because the core of the tumor is the area which the neurosurgeon remove. But what we are interested, the clinicians are interested in, are the margins of the tumor which are left behind, and then disseminated cells, which are mostly cancer stem cells. So those are the ones we want to target, but those are surrounded by tact plate barrier. So coming back here, the tumor left behind is representing the tumor boundaries where the barrier is intact. And this is very well exemplified by the disappointing efficacies of epidermal growth factor receptor inhibitors, which is the epitope of tyrosine kinase receptor targeting here. So in this diagram, where is red is where currently the small molecule inhibitors and other chemotherapeutics pull. So as we go from red to blue is the areas of intact blood-brain barrier where we have very low or no level of drug accumulation. And our goal here is to revert here, to make the red blue and the blue red. So the current solutions and current outcomes for these patients are basically based, on debulking radiotherapy and chemotherapy, and you see here the very dim survival. Combined with anti-angiogenic inhibitors like avastin, clinically used, and emerging small molecule tyrosine inhibitors, which failed to induce sustained response due to their low solubility and limited uptake by the tumor lesion with intact blood-brain tumor barrier, and of course high turnover of mutation expression leading to resistance. We already have chemotherapy-soaked polymer implantables known as gliadel, which is very toxic with very limited effectiveness. Currently in clinical trials, tumor treating electrical fields, and there is intensive ongoing research in terms of genome analysis. So we have ongoing clinical trials, phase 2 and 3, with combinations of different tyrosine kinase inhibitors, but they also face the same drawbacks in terms of solubility and delivery. Immunotherapies with dendritic cell-based vaccines and high-intensity field ultrasound, temporal disruption and delivery of liposomal drug carriers. So, based on previous observation where we could direct silica-coated nanoparticles to the nucleus of the cell in lung cancer studies, and following, this well-described methodology, we developed, theranostic, ferromagnetic, dye-doped nanoparticles complex to proprietary tyrosine kinase inhibitors. We had two different populations of nanoparticles, positively charged which are amino end fractionalized and carboxy end fractionalized, in order to assess the effect of coding and zeta potential in the delivery. And we used them in orthotopically planted tumors based on glioma stem cell-driven models. And as you can see here, we have been able to achieve a model recapitulating the in vivo condition where we have highly vascularized tumor, high cellularity and vascularization at the tumor borders. And we feel that it's a very good model, to test these particles. So why we think our solution is unique and has chances to work, what we call here unique selling proposition. We can achieve amplification of the effectiveness of targeted therapy via multimodal and multifunctional theranostic carriers. This way we can also speak of augmented reality in the surgical field and guided surgery. We can have accumulation at the tumor regions of high clinical significance. These are the borders, and where is red is our nanoparticle. Green fluorescent is green fluorescent protein transfected glioma cells. So it's obvious that we have accumulation at the tumor margins, where we have glioma stem cell niches and tumor boundaries. Furthermore, we have augmented biological activity via effective grafting and intracellular trafficking. These tumors express EGFR in the nucleus and in the mitochondria, so it's very important to see if we can, if we can achieve this, within the nucleus. So we try to see if we have nuclear targeting and delivery of specific pharmacological activity. And where there is suitability for long-term use, and this has been shown in basic clonogenicity nanotoxicological studies. And this particular drug, can have the potential for orphan drug designation and breakthrough designation because we have low prevalence of disease, and it's 3 out of 100,000, and its sufficiency of current solutions, as already described. So we can have favorable regulatory approaches and long-term exclusivity with this regulatory approach. So I can say we are currently at tier rank 3 towards 4, and the proof of concept here going all the way from in vitro studies where we documented nuclear targeting and downregulation of phospho-EGFR, where is green here is phospho-EGFR, which is downregulated in both cases. We use 2 different tyrosine kinase carrying subpopulations here. And going on to in vivo studies, what is really important, when comparing the carboxy and functionalized versus the amino and functionalized, we see a real statistical significance in the uptake at the tumor matrix of the negatively charged nanoparticles. This is way opposite to what we have seen so far in most publications, where in brain tumors and other cancers, because of the glycosylation and the glycocalyx of the pores of the vessels, we have higher predilection of positively charged nanoparticles. So we have questions to answer here. We do have favorable perinuclear and nuclear matrix uptake. We have amplified biological response, and we see here a targeting of emerging resistance markers such as the nuclear phospho-EGFR. We have target-specific pharmacological effectiveness. So the open questions here is how we can revisit nanodrug design and then differential role of zeta potential and the neo-vessel pore charge functionality here. But as you can see in this new model, as we grow from the center to the periphery, we have higher uptake with the nanoparticles versus the free drug. So next step, where we go, the only way to predict the future, I think, is to invent it. It's not mine, but I co-sign that. The way forward is next generation tumor nanopharmacology. As we speak, we have ongoing spatial temporal map for the evolution of the blood-brain barrier in the tumor. And we're moving towards precision medicine attributes in order to manage lack of precise imaging and subsequent ineffective quantification. And we apply bioinformatics to resolve this issue here. And moving on to assessment of endothelial retention. And we also consider convection-enhanced therapy to minimize systemic toxicity. And of course, we are seeking translational pathways and pilot lines without getting lost into translation. This can never be one man's island. It's a collaboration. And we thank, European Commission for the funding through IAPP grant. And my colleagues here, as you can see, in different aspects of their work by different teams. And of course, my students and postdocs. I know it's all Greek for you here, but as you know, nano is the Greek word for dwarf. So in a researcher's night, we had a very successful booth where it says that nanotechnology is where dwarfs do great. Thank you very much.